So in some of the previous videos, we've been talking about action potentials, and we'll talk a little bit about impulse conduction. So basically, all that stuff that leads up to the action potential, that's happening in one spot in the neuron. And so in impulse conduction, we simply need to take that and move it down the length of the neuron. And if you have an unmyelinated neuron, that impulse conduction has to travel the whole length of the neuron. That takes time. But in myelinated axons, we have the myelin sheath, that covering we talked about, and the gaps in the myelin sheath, the nodes of Ranvier. So in a myelinated neuron, the neuron or impulse conduction moves by what we call saltatory conduction, which means it skips the myelinated sections and only has to have that action potential, that depolarization, repolarization process happening in the gaps or the nodes, so it can travel much faster. So what determines whether the action potential forms? Well, that has to do with the synaptic potentials. Are they excitatory? Are they inhibitory? There's usually a combination of both. Both can be graded or different strengths. So excitatory, they're going to lead towards depolarization, make action potentials more likely to get to the threshold. Inhibitory, move towards hyperpolarization, action potentials become less likely. So we have to add them all together through summation to determine whether or not that particular neuron is going to go through depolarization and so on and have an action potential, and that impulse is going to travel then down that neuron to the next step. So we've talked about neurotransmitters a little bit when we talked about the synaptic cleft, but again, basically how this all works is neurotransmitters are going to be, when that signal comes down that neuron, it gets to that synaptic knob or that axon terminal, calcium gates are going to open, calcium is going to move in. That's going to cause the vesicles that contain the neurotransmitters to fuse with the membrane. They're going to release some of that neurotransmitter into the synaptic cleft. It's going to diffuse across the membrane. It's going to bind to the receptors, create a new electrical signal in the postsynaptic membrane. Now, to control the activity of the neurotransmitter, we don't want them to, once they're released, to be there for a long period of time, because that's going to lead to signals that we don't want or don't need. So we have three ways to regulate the activity of the neurotransmitter. Some of the neurotransmitters are going to be reabsorbed by endocytosis back into the presynaptic membrane and be reused. Some of them are going to simply diffuse out of the synaptic cleft. It's not an enclosed space, so it can't affect that synapse. And then there are enzymes in the synaptic cleft called, and they're named based on the neurotransmitter they break down, but they're all going to typically end in ASE. So like acetylcholine broken down by acetylcholine esterase. Finally, drug addiction. Neurotransmitters oftentimes can be used to explain how certain drugs work. Some drugs block certain neurotransmitters and stop signals, like pain signals, for instance. Some of them are going to mimic neurotransmitters and cause signals that wouldn't normally occur. But other drugs have completely different mechanisms, and some of these we understand and some of them we don't. Drug interaction is a very complex process, so we still have a lot of questions about why some individuals become addicted and others. We're starting to see that there is a genetic basis to that, and identifying specific genes, for instance, as well.